Thank you, Randy, for the lovely introduction, and welcome everyone. Tonight I'll be talking to you about my PhD project. We're attempting to use the gut microbiome for early cancer detection. I would like to take you on a journey into what it means to be a researcher in this field, and hopefully by the end, spark your curiosity a little bit. The gut microbiome is a microscopic ecosystem that lives within your intestine. But don't let the word microscopic fool you, because the gut microbiome is absolutely magical. Think of it as a giant city. Think of the gut lining and the blood vessels as the public transport, the roads, the bridges of this city. Think of services like digestion and immune system support as the essential services of this city. And just like a city is inhabited by many different people of many different backgrounds, the gut microbiome too is inhabited by bacteria, archaea, fungi, and viruses. And these all work together or in symmetry with each other to help maintain balance within this system. And this balance, we refer to it as eubiosis. And when an individual has a gut microbiome that is in a state of eubiosis, it usually is a healthy individual. But there are factors that can push this balance into unbalance. And we refer to this as dysbiosis. And prolonged states of dysbiosis have been linked with different diseases, including cancer. Now, research so far has focused on comparing the gut microbiomes of individuals who have cancer versus those who don't have cancer. But we know that the best way to fight cancer is to prevent it. And we also know that your body starts giving you clues that a change is happening before the change happens. So we thought it would be interesting to see if the gut microbiome can give you these clues. Essentially asking the question, can the gut microbiome give you signals for future cancer development? In which case, what do we need? To sample people's microbiome takes a lot of time, it takes a lot of money. And how do you sample individuals who will develop cancer but don't yet have a diagnosis? The answer, as always, lies in data. And in our case, a lot of data. First, we need a large group of people. Large enough where, while most people won't develop cancer, unfortunately, some will. Next, we need to sample their microbiome. And we get this information from their stool. We sample the thousands of DNA fragments of the bacteria that are present in your stool, and that is how we retrieve this information. Next, we need health, more specifically your cancer data. And finally, information on your lifestyle. What do you eat? How much physical activity do you do? And lucky for us here at NTNU, we have access to the HUNT study. For those of you unfamiliar with HUNT, it's a huge study that takes place every 10 years since it started in the 1980s. Maybe some of you have participated. And it collects a wide range of data from medical examination, lifestyle questionnaires, and even biological samples. So thanks to HUNT, we have 13,000 microbiome samples. And of these 13,000 microbiome samples, we know 2,000 have cancer. And we know this thanks to the Norwegian Cancer Registry. Now, the microbiome is a very, very, very popular topic in science. Especially in the last 10 years, it has become increasingly popular to talk about all it can do, essentially unlocking the mysteries of life. And while it is true that understanding the gut microbiome can help us better understand keys to unlocking diseases and mitigating diseases, we also need to be extra careful on how we interpret our results, because false conclusions tend to spread like wildfire. And the most important thing you need to know is that the methods that I use to analyze the data, the statistics that I use, matters. Why? Because depending on the method that I use, I get different results. What I want to do is ask my data the question, which microbes within our gut microbiome increase or decrease your risk of getting cancer. But why does this matter? Why can't I just use normal statistics and call it a day? Well, let me make an analogy using COVID tests. Do you remember when they rolled out those at-home testing kits and we were all shoving sticks up our noses for what felt like an incredibly long time? We quickly had to learn the difference between a false positive and a false negative test. If you had COVID and the test said you didn't, that's a false negative. If you didn't have COVID, but the test said you did, that's a false positive, as opposed to the test being accurate and giving you the true positive and true negative results. And the point is that the same applies for my data. If I ask a question, a statistical question that isn't appropriate for my data, or if I handle the data improperly, I get false positive and false negative results. False negative being that my data is telling me these microbes are absolutely not linked with future cancer, when in reality they might be. And false positive being that my data tells me these microbes absolutely are linked with future cancer, when in reality they might not. And a key aspect of microbiome data is that we are still in the process of developing these statistical tools. And we're doing this to avoid the risk of having false conclusions. So what we aim to do, is use the gut microbiome to find links for future cancer. What we have is the perfect data set to do this. But what we need is the appropriate statistical tools. And this is what I'm working on right now. And we have so far found some bacteria that have been linked or associated with future cancer. But association alone doesn't mean that these bacteria cause the cancer. And that's a very important point. The association alone will not tell us that these bacteria cause the cancer. There can be other factors like your lifestyle, your diet, that affect the bacteria and also affect the cancer. So that's what we're going to be focusing on in the future as well. We plan on zooming into the roles of these bacteria and their interactions with you, the host, to better understand the dynamics and their potential role in future cancer development. Thank you.